A Brief History in Microscopy is the first in a series of 3 lectures that I will provide for Unit 1. If we look back over time, for centuries, scientists have been fascinated with trying to better understand the world around us. As time has passed, we have slowly begun to unlock the secrets that the world has to offer. Yet to really appreciate where we are today, it's important to gain a little bit of perspective of the history of the past. While you can easily imagine that early biology was a simple act of visualizing and describing, what you may not know is that humans already had a very natural curiosity and an increased suspicion about the things that could not be seen. And if we first start with the scientist on the left, this is Girolamo Fracastoro. He was an Italian physician scholar, meaning he was also proficient in mathematics, geography, and astronomy. In addition, he was a poet. And as a very young man, he already became quite interested in the unknown. A little bit of lore: at the age of 19, he was already an appointed professor at his local university. But what he's most credited with was back in 1546, he proposed that epidemic diseases were caused by transferable tiny particles or spores that could transmit infection by either direct or indirect contact. And he believed at the time that these spores could cause disease even without contact over long distances. And in his writings, these spores, he often referred to chemicals rather than any living entities, though. But Fracastoro was clearly on to something, that there were entities that we could not see with the naked eye that could indeed cause disease and were interesting to study. And in fact, his theory remained influential for nearly 3 centuries before being replaced by the germ theory. Now, germ theory is widely accredited to another Italian, this time Agostino Bassi. In short, the germ theory of disease, also sometimes called the pathogenic theory of medicine, proposes that microorganisms are the cause of many diseases. And although highly controversial when first proposed, germ theory was later validated in the late 19th century. And so what does any of this have to do with microscopy? Well, really it was the invention of the microscope that allowed the germ theory of disease to be proven. Zacharias Janssen was a Dutch spectacle maker. He was associated with the invention of the first optical telescope and is sometimes also credited for inventing the first true compound microscope as early as 1595. It's believed that his invention was really an attempt to help his patients. As an optician, he was trying to make magnification even greater for those individuals with really poor eyesight. However, the origin of the microscope, just like the origin of the telescope, is a little bit of a matter of debate. If moving right along, though, to Anton Leeuwenhoek, you've probably heard of Anton van Leeuwenhoek. He was another Dutchman. He was also a tradesman and a scientist, and he's commonly known as the father of microbiology. Leeuwenhoek is considered to be the first true microbiologist. He's best known for his work on the improvement of the microscope and is credited as the first individual to observe and describe single-celled organisms using his handcrafted microscopes. He originally referred to the microbes he observed as animalcules, which we now refer to as microorganisms. But he was also the first to record microscopic observations of muscle fibers, bacteria, sperm, and even blood flow in capillaries. As time went on, more and more scientists began to use these early microscopes to study both plants and animals. In the late 1830s, Theodor Schwann reported the presence of cells in animal tissue. Matthias Schleiden also described the presence of cells in plants. And in 1855, Rudolf Virchow extended the cell theory and concluded that all cells arise from pre-existing cells. And so this is an important slide here, the 3 postulates of the cell theory. The first was that all living organisms are composed of 1 or more cells. The second postulate is that cells are the basic functional units of all living organisms. And the third postulate is all cells arise from pre-existing cells. And so these three postulates of the cell theory still hold true today. If we fast forward to today, the realm of microscopy has greatly expanded. We can now visualize cells and even small components of cells with greater clarity than ever before, and the technologies continue to improve. The study of cells is simply called cytology. Cells can only be viewed by a microscope, although the detail that you can see depends on the type of microscope that you're using. The use of a microscope is simply called microscopy, and cell size is measured in micrometers, and there are 1 million micrometers in 1 meter. And on this slide, you see just a brief comparison of the different sizes of cells and some of the molecules within the cells even. Cells can be as small as 7 micrometers, as large as 120 micrometers, and they come in many different shapes: flat, cylindrical, oval, irregular, etc. If we move on to the different types of microscopes that are often used in the lab, they can generally be separated into several different classes. One grouping is based on what interacts with the sample to generate the image. Either light, as we see in optical microscopes, electrons, as we see in electron microscopes, or some sort of probe, as in scanning probe microscopes. The microscope you are probably most familiar with and maybe have used already in some high school or advanced placement biology classes is with the compound light microscope. And this is pictured here on the left. It uses light to image the sample, yet has a limited ability to see the smallest pathogens and individual cellular organelles. As mentioned, another major type of microscopes are the electron microscopes. There's two types. Both the transmission electron microscope and the scanning electron microscope. These are considerably more expensive. They require much more expertise to operate, but they do allow for greater magnification. And here you see an image on the left of a transmission electron microscope in which it's using electrons to pass through the cell. It can magnify about 100 times greater than a light microscope, and you can see on the right this is an electron microscope image of cilia lining the respiratory tract, and you can see the detail now of individual components of cells rather than just the cells themselves. And lastly, here's an image of a scanning electron microscope. You're also using a beam of electrons. It's now instead the beam is reflected off the surface of the cell to provide a 3D study of cells. And so as you see on the image on the right, now you have a nice three-dimensional image of those same cilia lining the respiratory tract of a human being. And so I think the magnification here was 3,300 times, but it provides a very clear, detailed image of a particular component of the cell.